Hey everyone, welcome back. We are in chapter 19, slide 19. We finished talking about the ear, and now we're talking about the eye and vision. So to better understand how we see things, first we need to understand the overall structure of the eye. The eye has three layers. The outermost layer, the most superficial layer, this is the fibrous layer. The fibrous layer, since it's the outermost one, it needs to be protective. It protects and also provides attachment for muscles, specifically the extraocular muscles, which we'll recap in a minute. The whites of your eye, that's the sclera. The sclera is the white protective outer part of your eye. It contains collagen, it contains elastic fibers, which is good because don't poke it too hard, but your eye is resilient and is a little bit squishy and can stretch a little bit. The sclera is covered by a very thin layer, or the outermost layer of the sclera, I should say. The outermost layer is the conjunctiva. The conjunctiva is this thin outer layer, stratified squamous epithelium. As you know, stratified squamous is good for protection. This would be non-keratinized stratified squamous. There are goblet cells for mucus production so that you can capture any particles that get in your eye. That's how you produce that sleep, that eye gunk, basically eye buggers, to collect any particles that have gotten into your eye, and you get rid of that. There's also stratified columnar epithelium, since there are ducts for releasing this material. And so you have mucus, you have tears to help get rid of stuff that may have gotten into your eye. So the sclera, part of the fibrous layer, the whites of your eye. That's not the only part of your fibrous layer. Oops. Attached to the fibrous layer are those extraocular eye muscles. Remember that there are 6 different muscles that move your eye. There's the medial rectus on the medial portion of our eye. If let's assuming this is the right eye. So this is the medial rectus, controlled by the oculomotor nerve. The lateral rectus is controlled by the abducens, cranial nerve 6. Superior rectus, this one here, is controlled by oculomotor, cranial nerve 3, for moving your eye up. Inferior rectus is for moving your eye down, controlled also by oculomotor. Superior oblique is that one that loops through the trochlea and attaches at an angle. This is controlled by cranial nerve 4, trochlear nerve. Inferior oblique comes at an angle on the bottom. That's for extorsion, while superior oblique is for intorsion. Inferior oblique is also controlled by oculomotor. So 6 different eye muscles. 6 different eye muscles, 3 different cranial nerves that control them. And they attach, their tendons attach to that fibrous layer of the sclera. The sclera is most of the outer portion of your eye, but on the anterior portion of your eye, it's clear. The sclera is opaque, while the cornea is clear. The cornea is transparent, clear. It is avascular. In order for it to be clear, there can't be any blood vessels. So the cornea is avascular. It's curved, much like anything like a reading glass. It's curved so that it bends the light a little bit. The word for bending light is to refract. The cornea bends light. It's where light first passes so that we can focus on something. This doesn't actually do the focusing, but it starts to help with it. So here's the cornea in the front of the eye, curved portion. Here's another view, and it's cut away right here. Light shines through the cornea first before it goes through the rest of the eye. It shines through this part. The outer surface of the cornea is a thin, non-keratinized stratified squamous epithelium. The inner surface has where there's fluid. We'll talk about the fluid here in a minute. That's covered by simple squamous epithelium. The cornea, because it's important for refracting light, if there's an irregularity in the cornea, that can mess up our vision. If you're familiar with astigmatism, astigmatism is where you have an imperfection in the curvature of a certain part of your cornea. And that means if you have astigmatism in a certain part of your field of view, it'll look distorted. So if you have astigmatism, you probably know what I'm talking about. If you don't, try Googling astigmatism, what it might look like, and you'll see just like a field of view, a part of your field of view will be distorted. And there are ways to correct this with lenses. So cornea, sclera, that's all part of the fibrous outer layer. The middle layer, the intermediate one, is vascular. Lots of blood vessels. The vascular layer. Sorry, let me back up since I'm talking about blood vessels. The cornea, since it's avascular, how does it get nutrients? It gets nutrients from your tears, from your lacrimal glands producing tears. Tears contain a low amount of oxygen, but enough to pass it through. And also we'll see the fluid on the inside of the cornea that also provides oxygen. So the cornea still does get oxygen, but not from blood vessels directly. The middle layer, the vascular layer, tunic is another word for layer, represented here in this pinkish red. This supplies blood vessels and lymphatic vessels to most of the eye. Now, there are different parts that I'd like you to know that are specialized more towards the anterior side. If we look, let me use a different color. If we look over here in this cutaway, you can see this layer that goes around, and it would come all the way around, and it has a hole in it in the center. This is the iris. The iris is the colored part of your eye. It's colored, it's pigmented, so that it can absorb light. We want light to get into our eye through that hole, that pupil, but we don't want too much light, otherwise it'll just wash everything out. We don't want to get inundated with too much light. That's, if you open up too much light, just get blinded. We don't want that. So the iris does absorb some light, and people have different color eyes at this point with sunglasses, and it really doesn't, it's just kind of, what do you call it? It's just a genetic, a fun genetic trait, but the pigment does help block out sunlight. People with albinism, without pigment there, they're highly sensitive to light. So not only does it have pigmentation to absorb light, but the iris is also muscular. There are muscles, pupillary muscles, that control the size of your pupil. The pupil is the literal hole where light passes through. So pupil is not a physical structure, it's the absence of a structure, it's a hole. Pupillary muscles in the iris change the size of this hole, change the size of the pupil. It can constrict, and remember, constriction is controlled by cranial nerve 3 via parasympathetic pathway. And then also dilate it. We dilate our pupils with a sympathetic pathway. So this won't be through a cranial nerve, it'll be through a spinal nerve climbing up the sympathetic chain ganglion heading towards the pupil. Pupillary muscles controlled by sympathetic and parasympathetic pathways. The ciliary body, use a different color. The word cilia, we've seen this word a lot of times. Cilia means hair or hair-like. So what are the hair-like things? Let me erase this a little bit. It's more posterior to the iris. Look for these little processes. Those are the hair-like things, but the actual ciliary body is what surrounds it. So you can't see it here, but it's posterior, and it's all of this. I'll switch over to complete anatomy in a second to give you a better view of this. But in this view over here, it would be all of this. That would be the ciliary body versus the iris, which is more anterior and smaller surrounding the pupil. That would be the iris. The ciliary body contains ciliary muscles, and those little hair-like extensions attach to the lens. The lens is similar to a glasses lens, but the problem with glasses lenses is that they're fixed. They're hard. They don't change shape, not without, messing them up. Our lenses in our eye, it's a little bit squishy. It can change shape. The ciliary body, by attaching to the lens, can pull on it to stretch it out or relax it. Ciliary muscles control the shape of the lens, and that allows us to focus light even more than the cornea does. By having the lens make in different shapes, it changes the amount of refraction. Remember the term for focusing light? This is called accommodation. So ciliary muscles, also controlled by cranial nerve 3, oculomotor nerve, focus light by changing the shape of the lens. The rest of this vascular layer, everywhere else, is referred to as the choroid tunic. We've seen the word choroid before. We talked about the choroid plexus. That's in the brain and the ventricles. And plexus means a network of blood vessels. Choroid means blood vessels. Plexus means network of. Tunic means layer. So this is a layer of blood vessels. It's just a layer in that middle part of the eye. Highly vascularized and attaches all the way around and secures the retina, which we'll see here next. I'm actually going to switch to complete anatomy just to give you a better view of what we've just discussed, because I know it's a little bit hard to view. So if you take a look in the week's module, go down to videos, go to complete anatomy, that subheading, and you'll find whole eye model under complete anatomy. That's what I'm going to right now. I've limited it to the terms that I'd like you to know, because if you just go to the eye model that they have here, there's going to be a ton more terms a ton more structures that I'm not asking you to know. But here we can see some major structures. You can see the outside of the eye and the skin and the eyelashes. This is the superior view. This muscle right here, it's not superior rectus. This is the levator palpebrae superioris, also controlled by oculomotor nerve, cranial nerve 3. So that's levator palpebrae superioris. It raises your eyelid, your upper eyelid. Superior rectus is this one here. So it's just deep to it. This would be the left eye. We can see that this portion is medial. We can see a lacrimal duct here that drains into our nasal cavity. When you tear up, it can drain off your face, but it can drain in this duct that goes into your nasal cavity. Like if you cry, you get a runny nose. So that's medial. And so if that's medial, here's a medial rectus. Inferior rectus is down here. If we look underneath, so we're looking a bit underneath the eye, we can see the inferior oblique. And then let's look at from a top view again. Let me zoom out more. Here we can see the superior oblique. Superior oblique goes through that trochlea and allows it to attach at an angle. That's a superior oblique. All of these muscles attach to the sclera. Sclera, there we go. If we hide the sclera, we'll see the next layer down, and the one colored here in red is the choroid plexus, or excuse me, choroid tunic. If I hide this layer, I'll actually have to hide a few more things. There, now we can see inside. So we've basically cut the eye in half at this point. What we can see here, actually, let me undo what I just did. The layer that I had just removed and I'm adding back now. There we go. Okay, that's the retina. So we went from sclera, choroid tunic, and here is the retina, it's an innermost layer where you find photoreceptors. We'll get to this in a minute. I'm going to hide this for a second and show some more structures. I'm going to hide some of these muscles. If you look inside the eye's been cut in half. This is an inside view of the eye. Whoops. Inside view of the eye, do you see this texture right here in this circular? And it would go all the way around, but again, it's only half showing. This is the ciliary body. It's best seen from this posterior inside view. The lens would be right here. The hole where light passes through, there's a pupil. You can't really see the iris from this point of view. You need to go to the other side, the anterior view. So I'm going to clear this. Oops. And here you can see the iris. Oops. This colored portion of the eye is the iris, where you find the pupillary muscles to change the size of the pupil. The cornea is the clear curved layer which can best be seen from a side view. Let me actually hide this eyelid. No, I gotta hide the muscles as well. All right, a lot of things to hide, but can you see this really clear portion right here? That's the cornea. So we've seen a bunch of parts of the eye. Let's go closer and look at the retina. The retina, again, is all the way this deepest layer in the back. This is the retina. All right. Switching back to the slides. Slide 23. The retina, the innermost layer, contains photoreceptors and sensory neurons. We've actually seen this before when we discussed the optic nerve, but here it is again, and we're going in more detail. The receptors are photoreceptors. Remember, this is neuroepithelium. There are two different types of photoreceptors. There are rods and there are cones. They're named after their overall shape. Rods are kind of rod shaped. You can see one pictured down here. All cones are kind of cone shaped. You can see one pictured right here. Rods are adapted for low light. So they're used a lot at nighttime. It's also highly concentrated in your peripheral vision. So like I'm staring at you right now, I can still see stuff outside of here. It's not in the most focus, but I can see that like my hand is waving out this way. I can see it on this side, but I can't see all the detail. That's because my rods are allowing me to see that. Cones, they're your color vision photoreceptor. They let you see colors. If you are colorblind, there's different types of color blindness. The most common one is where there are actually 3 different types of cones, and if you're missing one of them, then you see a smaller range of colors. Like it's hard to distinguish between red and green in the most common type. That's a genetic condition. Cones are more concentrated in the center of the retina where we focus light. And so they allow us for sharp vision, for acuity. Acuity means really detailed, sharp vision. Rods, peripheral vision, low light. Cones, color vision, acuity. Pictured, like if we pictured right here. So here's a portion of the back of the eye. That's what we're looking at right here. This side is posterior. This side is anterior. And the arrangement of the photoreceptors and the neurons is actually kind of weird. Photoreceptors are in the back. So that's kind of weird because in order for photoreceptors to be activated, they need to detect light, but light is coming in from the anterior side and has to pass through all these layers of cells to get to the photoreceptors. So you're actually going to lose some quality here. Our eyes are kind of weirdly designed in that way. We would have better vision hypothetically if we didn't have all these cells in the way, obstructing our light. Photoreceptors, when they're activated by light, let me draw that again. If a photoreceptor is activated by light, the next cell in line to be activated is a bipolar cell. And a bipolar cell, as the name suggests, is bipolar shaped. It's very short. Bipolar cells then activate the next cell in line. It's called a ganglion cell. It's also bipolar shaped, and its axon extends out of the eye, which we'll get to in a second. Well, I'll just mention it right now. The axon, let's pretend this is a bipolar cell right here. Can you see my drawing? Here's a bipolar cell. Its axon bunches up and exits the eye in the back. The optic nerve then, the optic nerve are the axons of ganglion cells, just bunched up together in the back. On the right side, here's histology of, bottom right is a histology of the retina. You can see the choroid here in the back. This is where blood vessels are. An example of I can't tell the difference between rods and cones at this magnification, but you can see all these nuclei here. So let's say here is a rod. It's definitely not that big, but here's a rod. A rod then communicates with a bipolar cell, dendrites, axon, axon terminal. And then a bipolar cell communicates with a ganglion cell, dendrites, cell body, and then the axon goes out somewhere off to the side. Three important cells in line: photoreceptors, bipolar cells, ganglion cells. There's actually a couple other cells. We're not going to get to that. If you take a perception or neurobiology class, you'll learn more about amacrine and horizontal cells. So this is all found in the retina. More about the retina. I said that there's peripheral vision and then high acuity focused vision. When you focus on something, so when you look directly at something, so here's your fixation point, light bends a little bit, refracts a little bit thanks to the cornea, but your lens will refract the light even more so that you focus it on a single point. There's still light bouncing off to other parts of your retina, and you will see that light. You'll see that light that's been bouncing off of whatever objects you're in your peripheral view. This is your periphery. Where you have mostly rods. But where you have your focused light, this is mostly cones. Because cones are for high acuity. Even more interesting, if you look in more detail at this place, which, by the way, we call the macula lutea, where we focus light. We can see a high concentration of cones, lots of cones here. More interestingly, though, is that there's a little pit here that forms because the axons of the ganglion cells have been pushed and parted to the side. You can see this this ganglion cell, its axons coming this way, this ganglion cell, its axons coming this way, so we've created a little part. The term for this area is called the fovea centralis. Fovea means pit. If you remember fovea capitis, of the head of the femur, fovea just means a little pit. Fovea centralis. This is the center of the macula lutea, and it allows for the most focused color vision. It has fewer axons in the way, giving you the clearest picture. So that is why we focus light on that one spot. High amount of cones, fewer axons in the way. Because the axons of these ganglion cells need to exit somewhere out of the eye, so let's take a look down here. Because axons from the ganglion cells need to exit at some part of the eye, we bunch it all in one part. That bunched part is the optic nerve. And because you only have axons there, you can't have photoreceptors. There are no photoreceptors in this area, this rounded area. This area, it's somewhat near to the fovea, but a little bit off in the periphery. This is your optic disc. You might know this better as your blind spot. I'm really bad at doing this to myself, but look up on YouTube on how to test your blind spot. But if you stare straight forward and have a tiny object or point of something in your peripheral vision, you can kind of make it disappear for a second because there's no light actually hitting that one spot. we don't normally perceive our blind spot because our brain is good at filling things in. It's filling things in that's not even there. So our brain tricks us pretty much. But the optic disc, no photoreceptors because axons of these ganglion cells are exiting to form the optic nerve. We can divide up the eye into two basic cavities on the inside. And the inside is just as important for maintaining the structure. Here we have the cornea. And then here we have the lens. In between the cornea and the lens, this is the anterior cavity. It's filled with a water-like fluid. It's filled with aqueous humor. Aqueous means watery. Humor is an old term for fluid. So aqueous humor. Ciliary body cells secrete this fluid. So here's another function of the ciliary body, not just to cause accommodation of the lens, but also to release this aqueous humor. And it provides a little bit of pressure and it helps transport nutrients and wastes and cushion the cornea on the anterior side, or just posterior to the cornea. So it's really important for providing nutrients, getting rid of waste. If you are unable to drain, that leads to a buildup of pressure, and buildup of pressure for prolonged periods of time can lead to damage. Damage to your eye can lead to glaucoma, which is where you damage your cells and it can lead to partial or even full blindness. This aqueous humor, while it's created in this anterior portion, it can flow on these edges and also nourish the retina. So it does circulate throughout the eye. So here's its own mini circulatory system for the eye provided by aqueous humor. That's all in the anterior chamber and dripping into the posterior chamber. The posterior cavity, excuse me, anterior cavity versus posterior cavity. The posterior cavity is much bigger. It's all of this. Everything posterior to the lens is the posterior cavity. And instead of being filled with a watery fluid, it's filled with a very jello-like substance. I don't know if I should call it a fluid because it's very jello-like, very gelatinous. The term for this is vitreous body. You might see it called vitreous humor. I don't like calling it that because it's not very liquidy. It's got substance to it. So vitreous body. This gelatinous stuff helps support the shape of the eye. We have a nice rounded shape to our eye and it can still squish a bit. So yeah, provides shape. Here is an eye, it's a cow eye. Here's an eye that's been cut in half and you can see this jello-like stuff. That's the vitreous body. If you take a closer look, this is the back of the eye, the back half, the posterior half. Do you see this wrinkly stuff? That's the retina. The retina is not normally wrinkly. It's a thin layer and we want it to be flat. And to flatten it out, the vitreous body pushes up against it. Since it's been cut in half and the vitreous body's been removed, the vitreous body's not flattening it out. Think about like if you have a bed sheet and you're projecting a movie onto it, you want your bed sheet to be as flat as possible to get the clearest picture. If your bed sheet's wrinkled or folded, you can still see. It's just not going to be very clear or as clear as it could be. That's what could happen to our retina if we didn't have the proper amount of vitreous body. There's a condition where your retina can detach. If your retina detaches because of manipulated pressure of the vitreous body, if this shrinks or gets damaged, then that can lead to a detached retina. If you get hit hard enough, which yeah, don't recommend, can detach your retina and therefore affect your vision. Recapping what we learned back when we were learning about cranial nerves, about optic nerve. In the back of the eye, let me show you this part. Wait, no, let me show you this part. I'm showing you the medial portion of the left eye. In the medial portion, left eye, we have photoreceptors. The photoreceptor then activates a bipolar cell. Bipolar cell then activates a ganglion cell. This ganglion cell exits out the optic disc. Its axon forms the optic nerve. At this point in the pathway, we are here. Let me use a different, okay, yeah, that's good. Okay, so at this point in the pathway, we're right there. Then we have to make a decision. Are we going to cross or not? If we're on the medial portion of the left eye, we're getting information from our left visual field. Information from our left visual field is going to get processed in our right visual cortex. So there will be decussation here. Decussation will be happening at the optic chiasm. It's still the same axon, but we're crossing over. We end here in the thalamus. And then from the thalamus, we go to the primary visual cortex. The term for this axon, these are optic radiations. If we see something in our left field of view, it gets processed in our right visual cortex. There's contralateral processing of field of view. It is not exactly related to which eye. If I had drawn this for, let's do this in orange. If I had drawn this for the lateral portion of the left eye, it would be getting information from your right visual field. Right visual field gets processed in the left visual cortex. So if you send information, I'm just shorthanding here, it would not decussate. It would go this way to the visual cortex. There's contralateral processing of visual fields, but it differs based on the eye. I gave you 2 examples here, and hopefully you can figure that out for the other side. I suggest you practice drawing out these pathways. So to recap what we've learned in this chapter, we've gone over many different receptor types. And we see how our biological receptors, our biological receptors are a lot like receptors or detectors that we use in real life, or wherever we use everything in real life, but like these machines. If we want to detect metal, we need a special metal detector. If you want to detect hearing, if you want to detect sound, we need hair cells. If you want to detect skin stretch or skin pressure, skin light touch, skin grip, we need special receptors. If we want to detect pressure in our blood vessels, we need baroreceptors. If we need to detect smell, we need olfactory sensory receptors. If we need taste detectors, we need gustatory taste cells. we have very specialized things to detect very specialized stimuli. And there's many different types of stimuli that are useful for us to know about, whether it's coming from external things like light or sound, or internal things like blood pressure and chemicals in our blood. So that wraps up chapter 19. Lots of important videos, histology, models, dissection that we're going to practice in class that you can review. So I encourage you to look at those. As always, please let me know if you have any questions. There's questions in the comments in the discussion. And I'll see you all later. Thanks.